and today we will talk a bit about what are microbiota, what does it actually mean, and what are researchers doing research on actually to find out more about microbiota. And basically, microbiota are all the small organisms we have, for example, in our intestines, in our gut, but also some other environments we find them in. And we will just explain a bit why we find them so fascinating and interesting. But before we do this, I wanted to talk a bit about my background a bit more. I'm a biologist and I work as a researcher on a biological station called Nebius Svanhoft, which is located about 400 kilometers north of the Arctic Circle. And what's also very interesting about the station that we are basically located in this little wedge here between very close to the borders to Russia and Finland. And therefore, we're doing a lot of collaborations with our partners and neighbors on research, on Arctic and Nordic research. And here on the left-hand side, I just put a photo that you have a bit of an impression of the station. This is a photo from the summer. Right now, we have already a bit of snow with the botanical garden, a little pond in front. Just to drive really the point home how close we are to some of our neighbors, I put a photo here at the bottom. So this is the biological station. You basically can walk a bit, 50-minute walk, and you come to a river called the Pacific River. We're doing quite a bit of research in this river on fish and also microbiomes. And the middle of the river is also the border to Russia. So we are really, really close to that border. And our portfolio of research was in the beginning very connected to mammals, large mammals like brown bears. But over the years, we have expanded and doing all kinds of research on different organisms, from mammals to fish to insect species. And we often use genetic methods, which I will talk a bit about today. And a lot of this research is, of course, connected to climate change or climate gradients we have in this region. Today, I will talk a bit about first a bit about terms we are using in this context. What are microbiota? What are microbiomes? What is the Holubion concept? And then a few examples how we are using this and why this is interesting, why this is of practical interest also in terms of aquaculture and food security. And with this, I hope to give you a bit of an overview why research on microorganisms is so important. And if we first look at a few definitions to get started, then the term microbiota is very, very important. And microbiota is basically the entirety of microorganisms we find. We can find like bacteria, viruses, fungi, protists. And a biome, we generally refer to a habitat or ecosystem that has a specific set of flora and fauna, so a specific set of organisms like plants and animals. Let's say a forest biome has a specific set of organisms living in it. And if you merge then these two terms, microbiota and microbiome, or a biome, we receive a new term which is called microbiome. And micro is something small, in this case that we can't see as humans with our bare eyes. And the biome is then a specific location, basically, where we find those organisms, those very small organisms. And for example, here on the right-hand side, I simply showed a figure of a human, and we have, for example, a microbiome specifically to our intestines, to our digestive system, for example. And this has received quite a bit of attention in recent research because we can connect the state of the microbiome So if we have a healthy microbiome, we can connect this to human well-being, to immune responses, for example, but also behavior, memory, and a few other things. So this is in the medical field really research that is ongoing and has given some fascinating results about our cells and how our body actually works and how important microbial diversity is for our health. Talking a bit more about human microbiota and microbiome, it's probably interesting simply to get a few facts about this. So about 50% of your body cells consist actually of microbiota. Again, these are bacteria, virus, protists, and fungi. And what is also very fascinating is how high the biodiversity is actually. So in your gut microbiome alone, you have probably up to 5,000 different species. That's a lot of different organisms living in humans. And it weighs about 2 kilograms. And another very important point is that we as humans have, of course, a genome, a genetic code that codes for proteins and enzymes and so on in our system. And but all these different organisms living in us also have genomes. So it's estimated that there are probably about 200 million genes in those microorganisms we have in our body. And just in comparison, the human genome has about 20,000 genes. So what this means is that there's a plethora of genetic information also from different organisms in us, and it's basically second genome. And I will touch on this when I talk about a bit later about the Holubion concept. But first, I thought I explain why actually we are studying microorganisms right now. Why not earlier? Why is now the time that we are so interested in them? The short answer is that we are now able to study a wide variety of microorganisms at the same time and cost efficiently, and that we have now the technology to do this. And just to give you also a bit of a historical background, around 1600, the first microscope was invented. And with this, we were able to see some of the larger bacteria and other microbes, probably in the mid 1600s. Then we jump probably about 150 years later. It took 150 years to really realize that some bacteria or some microorganisms are connected to disease. So this is spurred the first interest really to study microorganisms even further. And also in the 1800s, basically the standard techniques to study microorganisms were developed. There were a few changes, few innovations, but more or less from this time onwards, how we grow, because we call this also culturing, and how we are dyeing them with a dye to better see some of the features. The microscopy field stayed more or less the same. There were, of course, some technical improvements in the microscopes that we could see smaller organisms and smaller particles. However, the principal concept of microscopy stayed the same. And one of the big problems with that was that we couldn't actually grow or culture all microorganisms in the lab. Some of them have so specific environmental conditions that we couldn't reproduce this in the lab, which meant that we are only studying a subset of microorganisms. However, with the invention of DNA sequencing, so determining the genetic code, it was then possible to also study those microorganisms with their genetic code, even if we can't grow them in the lab. This was really a breakthrough technology to really understand how diverse microorganisms are, and to understand a bit about evolution and understand also more about the genes, the functionality of the genes that microorganisms have. And if you're interested in DNA sequencing or genetics. I have also a presentation on that in the series for Interact. And just to give you a bit of an overview how we approach this in the lab. So let's say we go to this Pasvik River that I mentioned in the very beginning. Then we probably catch a fish, and we use also some of the tissue for other analysis, for example, looking at heavy metals and other things. But in this case, we are interested particularly in the intestine that we can separate and then extract DNA from all the content of the intestine. And then in the lab, we can determine the genetic sequences, genetic code of organisms that we find in the intestine. And just as an example, I have given here probably had two samples in the lab, and we find differences highlighted here in red. And from this, we can, for example, determine that we have two different microbial species or strains, and can then further analyze what does this mean? Are these new? Are they very different? What function probably the sequence has, and so on. And so I mentioned here and there about genetics also, and that the microbiome has basically its own genome. And this has led to the formulation of the Holubion concept. So as I mentioned in the beginning, you have, of course, your own, as humans, your own genome with the specific functionality of genes, but this could be also something else in nature. It could be a tree, for example. And then we have this extra microbiome in us, or it's connected to other organisms like a tree. And those genes can work together, they can interact somehow, and then change, for example, the function, or can enhance a function of genes or proteins. So there is an interaction between the genes of the microbiome and this host genome. And there are two different types. There are two different colors here. And basically, we have a part of those genes have co-evolved. They have changed with time to adjust to changes, for example, in the environment or other pressures. And then there's a type here shown in red that didn't undergo this long evolution, but still can contribute to changes and responses together with the host genome. And this is basically Holubion, the idea that the genes of microorganisms and either humans or other host species like a tree work together, that there's an interaction. And that's, of course, this was really very groundbreaking, that it's not just the genome of us humans, for example, that determine, for example, immune responses, but that also microorganisms that we carry in us can help with some of these immune responses, for example. And finally, we have sort of an environmental, what they call here environmental metagenome. This is simply other microorganisms that have no connection to this holobiont. They're just other microorganisms in the environment. And as scientists, we of course like to do experiments to actually test whether a concept or a theory could be proven to be true or to reject that theory. And indeed, this was done with the holobiont concept. So the idea was that in fish species, you have some fish that are more cold tolerant, so they survive in colder water more easily than others. So they're more temperature sensitive. And if this Holubion concept is true, then in those fish, you should expect a similar pattern. You should expect a change in the fish, but also in the microbiome if they live in different environments. And in the lab, this was actually tried out to breed a specific fish species with a specific natural gut microbiome. And then researchers bred them, selectively bred them to a population that is more host temperature sensitive. So they're more sensitive to temperature changes, and a more temperature resistant population. So they are not so influenced by water temperature. And the idea was then, okay, if there is a connection between the microbiome and the host, that you should see differences between these two populations. And indeed, this was the case. So that's why you have different colors here a bit. So specific microorganisms were found in each of these populations, and also the microbiome was different to the original population. So you had shifts in the number of species and which species were found in each of these populations. And with this, there was a strong suggestion that actually the Holubion concept is indeed true. And this gave, of course, rise to then further questions. What does actually affect the microbiome, and how does the microbiome affect then in this case, fish organism? And a lot of research has gone into what factors are changing the microbiome in fish. And first of all, you have factors that are influencing the microbiome, but on the other hand, you have also an interaction that the microbiome in turn influences processes in the host fish. And these are partially intrinsic factors, so factors that are host specific, like age or which sex you have, like females or males. Genetic background, genetic diversity in the host population plays a role. And then also factors like disease, stress can affect the microbiome quite a bit. And for example, nutrients or what state and what condition the host fish is. And then you have other temperatures. We talked already about temperature, water temperature, but it could be light. It could be water quality, clean water or polluted water, and also diet. And then, so these factors can affect the health and biodiversity in the microbiome. And if there are shifts, this can in turn change then how the host is able to receive nutrients, because the microbiome is involved in breaking down or processing food and nutrients, which can affect in the end growth. But then you have also other things like brain function. You could have, I mentioned already, immune responses. So the healthy microbiome will improve your resistance to disease, for example. And just to summarize what I have talked about here over the last few minutes, I just thought I summarize this one more time. So the microbiome is definitely involved in the tolerance to abiotic stress. So abiotic means external factors like water temperature and so on. It is involved in the protection against pathogens in connection to this, more generally to host immune responses if we encounter disease. There's an interaction with the host genotype, and variation in the host genotype can also influence the microbiome. So general growth and development is influenced by the microbiome, partially by nutrient acquisition, and the host environment plays also a very important role how the microbiome is shaped. From a more practical perspective, this is important because in fish, We have more and more aquaculture. So we are growing fish into sort of closed water basins. And because we are doing this, we want, of course, the fish to be healthy and have also good growth, because it is also a food source, food production. And therefore, a lot of research has also gone into understanding the fish microbiome and what may be affecting the fish microbiome in agricultural breeding facilities. And first of all, studies have found that aquacultural practices, like, for example, how many fish are in a water tank, what are those fish fed, for example, can influence the fish microbiome. Then the water quality, temperature, salinity, how much salt is in the water, the pH level, as well as other fish treatments that traditionally have been used in aquaculture, for example, antibiotics. So in the past, a lot of antibiotics have been used in aquacultural fish tanks to prevent disease. However, more and more research suggests that this can basically wipe out or severely impact the fish microbiome. And this was done to avoid infection from bacteria and pathogens. But more and more, because fish well-being is, of course, also a concern, people try to adjust these factors in order to help the fish to have a healthy microbiome. So it's also of practical importance, this research, to really improve conditions in, for example, breeding facilities. So we have talked a bit about fish and why this is so important, but I just thought I'd talk a tiny bit also about other microbiomes, just to have mentioned it. So also plants, for example, have microbiomes in different parts, can be the leaves, but also very, very important is the soil microbiome, which is basically often found around the roots. And again, they're sort of in the symbiotic relationship that these microorganisms can help with a nutrient acquisition, for example. So quite important that also in soil we have a healthy microbiome, healthy diversity. However, there are even other environments like freshwater and oceans that are important. So in this figure, I probably start here with the soil because we just talked about this. Microorganisms are also important for the breakdown of organic material, like dead animals, dead leaves, and during these processes, they are actually responsible for breaking down this material and releasing nutrients back into the soil that then plants can take up to grow again. So they are basically sort of natural fertilizers in the soil, another very important function. However, I thought I also mention, because it's often sort of forgotten, that we have also in the oceans microbes that do photosynthesis. So they're using light, UV light, to build organic matter. And during this process, they're producing also oxygen. And about 50% of the oxygen produced on Earth is actually produced by microorganisms in the ocean. So very important functions there. And at the same time, also in the ocean, they have a function of breaking down that material to release nutrients back in the system. And then we have also very important are microbes in agriculture. And I just thought I will talk about this in a bit more detail. Again, because to demonstrate a bit why we need these microbes in really practical context for our food security as well. So first of all, a lot of these processes and industrialization we have lead to emissions, and And those emissions in different contexts, which we'll talk about in more detail, can lead to shifts in different environments. The same that we can have shifts in the gut microbiome, we can have shifts in, let's say, soil microbiome as well. And usually this is a shift to less diversity. And with this, we are losing also, think about the genetic background there again, that we are losing also a specific functionality because genes code for specific functions basically. And we are losing this over time. And just to mention a few processes here is that, for example, land use, so which mammals are grazing, for example, or which crops we are planting can influence the microbiome in the soil. Then we are releasing, due to these industrial processes, quite a bit either wastewater or from agriculture fertilizers go into water systems, and the microbes there experience sort of an eutrophication, so a lot of nutrients are in the water, and this can lead to problems, for example, algal blooms. So again, you have a shift in these systems that are not always for the better. Then one thing we have here is rice paddies. And rice paddies are very important provider of food. So rice feeds about 50% of the human population, but those rice paddies contribute about 20% of the methane emission. And this methane is one of the greenhouse gases. So also those systems, our agricultural use has important implications for the climate. And again, shifts in the microbiomes. So we are changing the environments quite a bit, and we don't really always know exactly what we are changing. And I just want to summarize these because everything is a bit connected here, really. It's a nice analogy to think about what we talked about in the beginning about the microbiome in humans, and where more and more research suggests that more microbial diversity is connected to a healthy microbiome and a healthy host, a human. Whereas if we have problems for whatever reason with our microbiome, that this can lead actually to different problems, to an upset stomach, or we have skin conditions or other illnesses in humans. And the same can be said basically about ecosystems in nature. Here on the left-hand side, we have an example where we have quite a healthy ecosystem with lots of different plant and tree species, but also higher microbial diversity. And remember, I talked about some of these functions that, for example, microbes are important in soil because they are breaking down organic material to release nutrients. So these nutrients can be taken up by those plants, for example, and this leads in general to higher biodiversity, and of course more microorganisms means that also they have all these different genes that fulfill these different functions. On the other hand, if we have a less healthy ecosystem, then we often have less diversity in the microbial community. So some of this organic material is actually not broken down. So these nutrients can't be released into the soil again. And in this example, it was a catastrophic event like a fire, which means that actually the ecosystem can't recover as quickly from this catastrophic event. And this means also in the long term that we probably as humans need to intervene more with management, like removal of specific plant species that that shouldn't be there, that are invasive. These are species that would not grow usually in this environment. And we have also talked about invasive species if you're interested in this series. And overall, you have it's less functioning ecosystem. So something to think about really about how important microorganisms really are in a lot of processes on Earth. And with this, I just wanted to close with some final notes. What I have presented here is really just the beginning. We are just starting to understand the interactions of the microbiome and external and internal factors, but also the interactions between the microbiomes and their host species. Right now, we are very much at the point that we are describing the diversity of microorganisms in different environments and other organisms. But the future will hold that we really try to understand those interactions. So it's a very interesting research field, and it's really expanding research field as well. And with this, I thank you for your attention, and I hope you have enjoyed this presentation. Bye-bye.